Okay, great. I think we are all a go, and we'll probably get started in just 1 minute. I always like to give people a little bit of time to come in, but I also want to make sure that you have the maximum time to speak today.
Yeah, YouTube looks good to 10, so I think we're done.
All right, thanks, Adam. I wonder how many more large pieces of machinery are going to come to Celeste's house in the next hour.
The street cleaner is going behind me now.
Are you close to campus?
About a mile from campus, yeah.
Nice. I love it there. I think Princeton just has the most beautiful campus. My sister's down there in grad school, and her first year grad dorm, it was like Hogwarts from Harry Potter.
It is, yeah. The dining hall looks exactly like how where they dine in Harry Potter.
Yeah, it's amazing. It's really wonderful. So
they live in castles here.
Until they move out and get apartments, and then it's like, okay, this is regular life again. Yeah. All right. I think, it's going to be a steady stream of people coming in over the next few minutes, but like I said, I want to give you max time. So I'm going to go ahead and get started. So welcome everybody. Good morning or afternoon or evening, whatever time it is for you. Thanks everyone for being here. Today, we are very thrilled to welcome Professor Celeste Nelson as today's speaker. Dr. Nelson is a professor of bioengineering and also professor in the departments of chemical and biological engineering and in molecular biology at Princeton University. She earned her bachelor's degrees in chemical engineering and in biology at MIT, and then a PhD in biomedical engineering from Johns Hopkins, followed by postdoctoral training at the Lawrence Berkeley National Lab. Dr. Nelson's lab at Princeton specializes in using engineered tissues and computational models to understand how mechanical forces direct developmental patterning events during tissue morphogenesis and during disease progression. And then just on a personal note, I want to say that Celeste is one of my science heroes. I remember discovering her work when I was sort of an early starting out graduate student and really being awed and inspired, seeing the way that she was using bioengineering strategies to answer really interesting biological questions. And so her work's been a big influence on me over the past years and probably a lot of you as well. So I was really happy when she accepted our invitation to come and speak here, and we're delighted to have her here. So Celeste, the floor is yours. Take it away. Thanks so much.
Thank you. That was an incredibly kind introduction. I've really enjoyed watching these seminars, and I'm so grateful that you put these together. And I'm thrilled to be able to share, that's not exactly what I wanted to do. Thrilled to be able to share my group's work with you today. So what I'm hoping to do today is to give you a slightly different perspective on cell migration and motility than we've heard from some of the previous seminars, by focusing on shape changes that happen within the epithelium as it builds into three-dimensional tissue structures, and some of what we've uncovered in my group about the roles of mesenchymal migration in that process. So before I get started, I need to unplug my mouse because Zoom and my mouse don't like each other so much. So that being done, where we need to begin is with the tree of life. So we've heard a variety of really diverse talks in this seminar series using a crazily large number of different model systems to investigate how cells move with respect to each other, how that's controlled, and what the implications are for our bodies and the tissues within them. I'm inspired by those different model systems, but mostly from the perspective of the different types of tissues that have been generated over the course of evolution. And in particular, I'm inspired by the different types of tissue structures that have evolved to permit oxygen to get deep within our tissues, to promote the generation of ATP that fuels these migratory processes that we're all so very excited about. And so I'm speaking in particular of the gas exchangers that have evolved over millennia to give rise to this beautiful ability to bring oxygen into our tissues. And these can be as simple as the body surface of organisms like flatworms that you see on the left, or they can be incredibly complex, like the tracheal systems, the tubes through which gases flow in insects, or the gills of the fish, which are very efficient gas exchangers. Today, I'm going to tell you a little bit about what we've uncovered with respect to how gas exchangers are formed in the context of terrestrial vertebrates, focusing on the lungs and on the lungs across the classes of vertebrates that have evolved to populate our planet. And so one of the reasons why I'm so inspired by gas exchangers like the lungs is because there's an amazing diversity in their architectural forms and the anatomies that have developed to permit oxygen to be brought into our tissues and waste products like carbon dioxide to move out of our bodies and into our ambient surroundings. And so two different forms of lungs are shown on this slide. So you should be seeing a GIF that shows the movement of fresh air deep into the lungs, and then exhalation of stale air out of the body cavity. And so when you and I breathe, we breathe using the mechanism on the top of this GIF. We contract our diaphragm, and if we're taking a really deep breath, we also contract the muscles in between the ribs and our rib cage. This increases the volume of our chest cavity and allows air to move from our nasal passages through our windpipe or trachea, and then into the primary bronchi that feed into ever smaller branches to eventually reach the gas exchange surface of the lungs. And then when we exhale, that stale air goes back through the same conduits and moves out our nasal passages or our mouth. And what this setup permits is a large surface area by virtue of the highly branched architecture of this structure, and a very thin surface to enable oxygen to diffuse in one direction down its concentration gradient, and CO2 to diffuse in the opposite direction down its concentration gradient. And so this, of course, is how we breathe as mammals, but it's not the most efficient gas exchange structure that has evolved. That would go to the birds. And so a model bird, a chicken, is shown on the bottom of this GIF, which illustrates on the left the very different anatomy for breathing that birds have. So they don't have a diaphragm, and instead they breathe by virtue of these air sacs, which are labeled in this GIF. So these are appendages off the surface of their lungs that act as bellows to permit the convection of air that then goes in a single direction through the gas exchange portion of the lungs that then allows for a higher concentration of oxygen to be present on the air side of the gas exchange surface than can be achieved in this biphasic tidal structure of the mammalian lung. So my group is really interested in understanding how these architectures form and what lessons we can learn from their morphogenesis to help us as engineers come up with strategies to build tissues outside of the body. As you might imagine, the development of lungs has really been investigated quite heavily over the years, given their importance to our everyday lives. And both developmental biologists as well as geneticists have uncovered a large number of pathways that are absolutely required for the formation of this tree-like structure that we have in the mammalian lungs. And this process that builds the tree is known as branching morphogenesis, where a very simple epithelium is sculpted over time, over multiple rounds of branching to generate that final arborized, ramified architecture. And what those developmental biologists and geneticists have uncovered is a huge list of genes that are absolutely required for the formation of the epithelial tree. that is important for breathing within the mammalian lungs. So you can think of this as sort of like the ingredients list that is necessary to build a lung. But these ingredients don't tell us anything about how we would go about forming one ourselves, or indeed how the lung is built itself from its resident tissues during morphogenesis. And so my group has been trying to ask sort of an orthogonal question to the conventional developmental biology approach, and that's, how are these tissues actually sculpted physically? And what I mean by that is, if we put that ingredients list to the side and simply imagine the tissues themselves as materials, say as like a simple elastic sheet that has an initial architecture. That elastic sheet or that material has properties, so it can be purely elastic or have some viscoelastic character to it. But regardless, all of those ingredients on the previous slide have to change the properties of that sheet so that it can fold or be bent, be expanded or be compressed or twist to ultimately form the branches essential for conduction of air through the airways of the lung. Or indeed, if we take a step back and imagine any tissue, to form into the organs that are essential for life of an organism. And so purely viewing these tissues as materials, then applying forces on them, physical forces on them, will allow a simple structure to eventually be molded into the more complex architectures that we see within the organs of our favorite model organisms. And so diving a little deeper, the key questions that we're trying to ask are, what are the physical forces that are responsible for turning those simple epithelia into these complex architectures that we see in the lungs of different vertebrates? And are those physical forces conserved across species? And then even deeper still, what are the cells, what are the populations of cells that are responsible for actually exerting those forces to fold those simple epithelial sheets into the complex geometries that we need in order to breathe after birth? And so if we think about what could be taking place to build these complex tree-like structures from a simple epithelium, there are a few possibilities that are shown here on this slide. So the first is that we have a sheet of epithelial cells that actively folds to generate a branch. And then the second possibility is that the sheet of epithelial cells is purely passive and responds to physical forces exerted either from its apical surface, so from the surface that will eventually give rise to the lumen that's filled with air in the case of the lung, or forces from its basal surface, from the mesenchymal population that surrounds this developing epithelium. Either way, that those forces exerted on a passive material could lead to a deformation that would permit the formation of a branch. Now, given that this is a community interested in migration and motility, you might be wondering why I don't draw on this slide a cartoon of the epithelial cells actively migrating into their surroundings to generate a branch, which is certainly how, say, the vascular system builds itself, or the trachea builds itself in the case of the insect breathing apparatus. The reason why I don't draw that in the context of vertebrate airways is that there's absolutely no evidence that the epithelial cells are migrating into the surrounding mesenchyme to generate a branch. They do seem to maintain their fully epithelial sheet-like behaviors. And so what I'm going to tell you today are 3 short stories focused on how these epithelial tissues are constructed into branches. And I promise you will see the role of migration here, despite that disclaimer I just noted on the previous slide. I'll start by telling you a little bit about what we've uncovered with respect to how mammalian lungs, the ones that we hold most near and dear to our hearts, are constructed. And then I'll tell you a little bit about what we've learned with respect to the evolutionary diversity in the vertebrate lung by focusing on chicken lungs. And then at the end, I'll show you some unpublished data that we're super excited about, describing how the reptilian lung builds itself. The mammalian lung is known as an alveolar lung because the functional unit of the lung is the alveolus, which is this really thin sheet of cells located at the terminal ends of the tree-like structure. The work that I'm going to share with you was initiated by several previous postdocs in the lab, and most recently is being driven by a very talented graduate student, Katie Goodwin. So just to bring you up to speed with respect to what the mammalian lung looks like, our model system here is the mouse. On this slide on the left, what you should see is a time-lapse video of an embryonic mouse lung that's been taken out of the embryo and cultured in a stage-top incubator on a microscope. And so you should see the lung undergoing branching morphogenesis to build additional branches over the course of the time lapse. And so just to orient you, this particular lung is situated so that at, I guess, the northeast corner of the organ is the trachea. That then splits into the primary bronchi that feed into the left lung and the right lung. And as you're watching this, what you should notice is that when the video loops again, that the epithelium itself is branching into daughter branches over the course of the time lapse. And so the light gray color through this organ is the epithelium. It's surrounded by a darker gray color, which is the mesenchyme. And then the entire surface of the lung is surrounded by an even darker gray color, which is a single layer of cells known as the mesothelium, which gives rise to the pleural surface of the lung. Most of the branches that are formed during morphogenesis of the mammalian lung are this bifurcating branching, where the tip of a parent branch splits in half to form two daughter branches. But the early structure of the lung is established by another process known as domain branching, where the parent branch forms daughter branches off of its side laterally in specific domains. And so this process of branching morphogenesis in the mammalian lung, particularly in the mouse lung, has been investigated, as you might imagine, for decades. We know that the process is highly stereotyped, meaning that every branch that forms does so in a predetermined location with a remarkably relative diameter and angle to the parent branch that is conserved across individuals within a species. And the architecture of the lung is so incredibly stereotyped that each of these branches has a name. They've been named by the first author of this paper that I'm showing on this slide. And so, as I said, the majority of the branches that form are these bifurcating branches in character, where the tip of a parent branch splits in half to form two daughter branches. And that happens over the course of about 23 generations within the mouse lung, which gives us a total number of terminal ends of 2 to the 23rd power by virtue of the fact that this is a bifurcating system, or approximately 8 million branches. And so we're really interested in understanding physically how those form, and then how that robust stereotypy is established in this system. So how do you have 8 million precisely determined branching events? What we found is that the epithelium itself appears to be passive in the mammalian lung, and instead is sculpted by the mesenchymal cells that surround the tissue, which are developing at the same time as these branches form. And the most prominent mesenchymal cell type that's differentiating during the formation of these branches is the airway smooth muscle, which wraps circumferentially around the epithelium on the basal side of the epithelium, and is derived from the undifferentiated mesenchyme that surrounds the epithelium as it's undergoing branching. And we sort of landed on this concept of smooth muscle playing an important role in morphogenesis somewhat serendipitously through time-lapse experiments that were being conducted for a completely different reason of lungs that were taken from embryos from mice that express red fluorescent protein downstream of the alpha smooth muscle actin gene promoter. And so alpha smooth muscle actin is a protein that's expressed in many cell types, including smooth muscle. But at this stage of development in the mouse lung, alpha smooth muscle actin is only expressed within the airway smooth muscle. And what we found, again, quite surprisingly and serendipitously, was that new smooth muscle cells differentiate at the branch site of the epithelium prior to the formation of that bifurcation each and every time we viewed these samples over the time lapse. And so what you should be seeing on this slide is a video of one branch undergoing a bifurcation on the left in the bright field channel. And then the smooth muscle is labeled in red on the far right and has its own channel in the middle. And I hope what you appreciate is that there's a bright spot of RFP expression at the future site where the bifurcation is going to take place before the epithelium has changed shape. And so that's summarized on this slide here. So what we found using a variety of different approaches is that the pattern of differentiation of this mesenchymal tissue is absolutely required for the formation of those bifurcations, and more recently is also required for the formation of lateral branches, or those domain branches that establish the initial architecture of the lung. And so our conclusions from this work are that the folding of this passive epithelium is shaped by forces from that smooth muscle that's differentiating in a very precise pattern around the epithelium itself, which then begs two questions. What specifies this pattern of smooth muscle? That's absolutely essential. And then in order to answer that, the second question is which cells are actually becoming smooth muscle in the first place, which surprisingly in 2021 still has not been accepted in the field. And so to get at the second question, Katie decided to carry out a single cell RNA sequencing analysis to start to try to uncover which cells are becoming airway smooth muscle within the developing lung. And so she took lungs from mouse embryos, put them into a single cell suspension, and then carried out single cell RNA sequencing with the hope of identifying the population of cells within the mesenchyme that is giving rise to the smooth muscle that could help us figure out how we achieve this robust stereotypy with formation of the branches within the epithelium. And so she found the cell types that you would expect at this stage of development. And so she pulled out epithelial cells, shown in blue here, smooth muscle cells, the population we're interested in, shown in the purplish color. She pulled out mesothelial cells, so the cells that line the surface of the developing lung, as well as vascular endothelial cells. And then somewhat surprisingly, two somewhat separable populations of mesenchymal cells that were closely associated with the smooth muscle itself. And so she thought, well, maybe one of these two populations is the population that gives rise to the smooth muscle that surrounds the airways, and would be the population to focus on in trying to understand the forces that are involved in formation of those branches themselves. And so what Katie did then is to recluster all of the cells that are in this red dotted box here, so the two mesenchymal populations and the smooth muscle population, to identify what the smooth muscle progenitor might be based on gene expression patterns. And she somewhat surprisingly found that when these are reclustered, they all sort of merge together. And doesn't appear to be a distinct progenitor population, at least based on gene expression patterns within the developing lung. And so then Katie went and asked, well, if these two mesenchymal populations, these two mesenchymal clusters, are both closely associated with smooth muscle, then what is distinguishing them from each other? Are they located in different regions? Do they have different functions? To look at the first question, what Katie did was to examine the different markers that are expressed in those two different mesenchymal populations, and then localize them to the developing lung using immunofluorescence analysis. And what she found is that indeed these two different mesenchymal clusters are located in different positions within the developing lungs. So one cluster, which expressed a unique set of markers, including very high levels of this marker LEF1, was located, as you can see in green in the top portion of this slide, in the mesenchymal cells immediately adjacent to the epithelium, which you can sort of see as the blue labeled cells in the image on the left, which is a nuclear label that is absent from the grayscale image of LEF1 on the right. And so we classify these cells given their location adjacent to the epithelium as the subepithelial mesenchyme, the subepithelial mesenchymal cluster. The other mesenchymal cluster expressed, again, a unique set of markers, including very high levels of FOXP1. And Katie found that FOXP1 is expressed both within the mesothelium that surrounds the lungs, as well as in the mesenchymal cells that are adjacent to the mesothelium. So these are then the submesothelial mesenchyme. So these two clusters are in distinct locations. And what I'll show you is that they also have distinct functions. And so Katie found that the subepithelial mesenchyme is the population that gives rise to airway smooth muscle. And the submesothelial mesenchyme gives rise to other mesenchymal populations, but in particular, the data suggest vascular smooth muscle. So I'll focus on the conclusion at the top, the airway smooth muscle conclusion. And this is where we start talking about the role of migration in development of these epithelial tissues that don't appear to migrate themselves to form the tree. So how Katie came to these conclusions were using a number of approaches, one of which was simply labeling individual cells and then tracking them over time to see which cells eventually located themselves within the smooth muscle layer and then expressed smooth muscle markers. And she did that by generating a mouse that expresses confetti. So the brainbow mouse expresses 4 different fluorophores under the control of derma-1 Cree. So derma-1 is another name for twist2. It's expressed really highly within the mesenchyme, but not expressed within the epithelium. And so these derma-1 confetti mice that Katie generated express 4 different fluorophores at random levels within the mesenchyme of the lungs. And so what you should see looping over and over again on this slide is a time lapse of a derma-1 confetti lung. You can focus on the bottom right corner, and you'll note that the epithelium is expanding and then forming a bifurcation during this movie. And the mesenchyme itself is highly motile during this folding process of the epithelium, which I hope you can appreciate by then focusing on one of the other 3 different panels, either the RFP panel, the YFP panel, or the merged panel of the 2 chosen fluorophores here. And so this mouse was really useful for uncovering which population of cells, either the subepithelial or the submesothelial mesenchyme, give rise to that smooth muscle population that is so important for formation of the branches. So what Katie did was to go in and use these movies to track the motions of individual cells, depending on their position, adjacent to the epithelium or adjacent to the mesothelium. And what she found is that she could track, by virtue of the fact that there were 4 different fluorophores expressed at different levels within each individual cell. She could track the different cells and then determine their motility characteristics. And so some of that is shown here. So there's stills from time lapses on the left. The middle panel here shows some tracks, some representative tracks of cells within the smooth muscle layer, within the subepithelial mesenchyme, or within the submesothelial mesenchyme. And then the graph on the right shows the average speed of the cell populations in micrometers per hour over the course of the time-lapse analysis. And what Katie's data show is that the subepithelial mesenchymal population is really highly motile, especially compared to the submesothelial mesenchyme or the smooth muscle, which doesn't seem to move around all that much. What she could also do then was to track whether cells stay within their compartment. So do they always stay within the subepithelial mesenchyme? Do they always stay within the submesothelial mesenchyme? Or do they move from one compartment to the next? And what she found by tracking individual cells within the different initial compartments was that some submesothelial cells, so shown in this top row here, move into the subepithelial location within the lung. Very few subepithelial cells move into the submesothelial location, but several subepithelial cells move into the smooth muscle population. And so the different trajectories and subset of populations that carry out these different movements are shown in the pie charts on the bottom. And so the conclusion then from all of this work, as well as from some genetic manipulations and pharmacological manipulations, is that the subepithelial mesenchymal population gives rise to the smooth muscle, which makes sense because it's located adjacent to the epithelium in the right spot to receive signals to differentiate into smooth muscle, but that there's fluidity within the mesenchyme. And the cells that are located adjacent to the mesothelium, for instance, have some propensity to mix into that layer. So now we know which cells become smooth muscle. And now the next question to answer that we're working on is what specifies that pattern of smooth muscle differentiation? And I think that now that we see how highly motile the mesenchyme is, our approaches for addressing that first question about pattern have to take into account the fluidity of the mesenchyme, the high motility of the mesenchymal cells. So in terms of the forces that induce the formation of branches within the mouse lung, our data suggests that the mouse airway epithelium is pushed, or it feels physical forces from the mesenchyme, specifically from smooth muscle cells that have differentiated from mesenchymal populations adjacent to the epithelium itself. And now we want to understand what directs the migration, the motility of the mesenchymal populations within the developing lung, and how are these patterns specified over time scales as well as length scales to permit the folding of that epithelium into its final geometry. So really what began as a very epithelial-centric question about how branches form has turned into a very mesenchyme-centric mechanism that we're trying to investigate. So that's what we've uncovered for morphogenesis of the mammalian lung. I started by telling you that there's an enormous diversity in gas exchangers, and mentioned that the avian lung, the bird lung, is much more efficient than the mammalian lung in getting oxygen into tissues by virtue of its anatomy, by virtue of its design. And so some of that is shown on this cartoon. The bird lung does not have alveoli. Instead, gas exchange takes place within air capillaries at the ends of, or that are dotted along unidirectional flow tracts known as parabronchi. And so what we wanted to understand is whether the mechanisms we were uncovering for physical forces exerting inducing morphogenesis in the mammalian lung are conserved across this structure. So these parabronchial lungs permit cross-current gas exchange and permit unidirectional flow of air. And so several previous postdocs, as well as a current graduate student in the lab, Michael Palmer, have been focused on trying to understand the physical forces that are necessary for building these lungs. And we use the chicken as our model system, the chicken embryo as our model system. Now, in contrast to the mammalian lung, the majority of the branches that form the entire structure, the entire tree of the bird lung, arise by this process known as domain branching or lateral branching. But just like in the mouse, the positions at which these branches form are incredibly stereotyped, so conserved across individuals of a species at these earliest stages of development. Just as with the mouse lung, the early bird lung starts out as sort of a wishbone-shaped structure where you have a trachea at the top. The epithelium in this cartoon on the right of the slide is shown in the orange color. And so the trachea is at the top. It bifurcates into two primary bronchi that feed the left lung and the right lung. And then branches form off of the dorsal side of that primary bronchus at specific positions and specific time points during its development. Now, amazingly, there's no smooth muscle present within the developing bird lung at this stage. And so that tells us already that there has to be a different mechanism for forming branches. And what we uncovered a few years ago is that the epithelial cells that are located at the sites at which these branches are going to form undergo a change in shape during the process. They go from being highly columnar in geometry to trapezoidal in geometry, where the short end of the trapezoid is located on the side of the epithelial cells that's adjacent to the lumen where air will eventually form, the apical side of the cells. And that change in shape is classically known as an apical constriction. So it's an active process that requires actomyosin-driven contractility at the apical surface to promote that change from columnar to trapezoidal morphology. And so we found this by looking at the shapes of individual cells as well as locations of contractility markers. And so what I'm showing you on this slide at the top is staining for filamentous actin using phalloidin. The trachea would be close to the title of this slide in panel A, in the F-actin panel on the left. The ventral surface of the primary bronchus is sort of facing like the southeast corner, and the dorsal surface is facing the northwest corner. And two branches are being formed at this time point in development, labeled as B1 and B2. And I hope you can appreciate the really high intensity of phalloidin stain at the apical surface facing the lumen of these two branches, B1 and B2. I hope you can also appreciate that in panel B, there's a really high intensity of phosphorylated myosin light chain staining, again, at the apical surface of these branches. So we think that active changes in cell shape are driving morphogenesis of this tissue by virtue of these of the staining profiles here, as well as the fact that if we inhibit actin contractility, inhibit apical constriction, then we block the formation of new branches in lungs explanted from chicken embryos. Apical constriction also appears to be a mechanism that builds lungs of other species. And so we've looked at the chicken, of course, shown here on the left. So that's Gallus variant domesticus. We looked at Japanese quail, shown in the middle. These are both in the same order of birds. And we've also examined embryonic lungs from Pekin duck, shown on the right, which is a related but different order of birds. And so this appears to be a conserved mechanism. So you might be wondering, well, how does migration and motility come into play if the epithelium itself is actively changing shape but not invading into the surrounding mesenchyme? Well, once the changes in cell shape have generated that branch, the branch then needs to extend into the surrounding mesenchyme. It appears to do so not through migration of the epithelium, but through physical changes to the mesenchyme that permit extension to take place. And so we come to that conclusion by looking at changes within the mesenchyme as well as within the extracellular matrix that surrounds the epithelium and that's present within the mesenchyme. And so shown here in the cartoon on the left are representations, so schematics illustrating what the epithelium looks like before it's formed a branch. So it's at embryonic day 4 in the chick, so E4. And after 3 branches have formed at embryonic day 5, when we stain for markers of basement membrane, which ensheath the epithelium at these stages, we noticed that the basement membrane is fully intact as the epithelial cells are undergoing that apical constriction. But after the epithelium has initiated a branch, the basement membrane is depleted at the leading edge of the advancing, extending epithelial branch by virtue of staining for laminin or other markers of the basement membrane. At the same time, mesenchymal ECM proteins appear to accumulate adjacent to the leading edge of the developing branch. And so one particularly compelling example is tenascin C, shown here in green on this slide, again, adjacent at the early stages and the extending stages of an extending epithelial branch. And the curious thing about tenascin C is that it's a huge protein. It's about 200 kilodaltons on average. And it's we see that it's located, several cell diameters away from the extending epithelium within this tissue. But the protein itself is only synthesized in the epithelium. So we have this huge protein, this huge extracellular matrix protein that is being made within the epithelium, but ending up distant from the epithelium during this process of morphogenesis. We have data to show that this extracellular matrix remodeling is essential for extension of the branch, elongation of the branch. And so then the question became, well, how does tenascin C get transported so far within the mesenchyme? And the answer was the motility of the mesenchymal cells themselves. And so we uncovered this by looking at the displacements of beads that we injected into the mesenchyme at different locations within the developing chick lung. So some of that is shown on the left here. So beads that were injected in the mesenchyme adjacent to epithelium that is not branching don't move very far, whereas beads that are injected in the mesenchyme adjacent to the epithelium that's extending are displaced quite dramatically. And so this, as well as some other pieces of evidence, told us that the mesenchymal cells that are located adjacent to the leading edge of these extending branches are moving dramatically. And the movement of those mesenchymal cells is what's responsible for transporting this epithelial synthesized protein deep into the mesenchyme itself. We think that this increase in motility is a result of an unjamming transition in the mesenchyme. And so we came to this conclusion by using snapshots of fixed specimens in which the individual cells could be visualized by staining for F-actin again, so using phalloidin, and then pulling out the aspect ratios and the shape factors of the cells within the mesenchymes, some of which is plotted here. But if you focus on, I guess, panel G in this particular image in the middle of this slide, the aspect ratios of the cells are color-coded. So cells that are more elongated, which is characteristic of more migratory cells and an unjammed tissue, are in hot colors, whereas those that are less elongated, so closer to circular in cross-section, and thereby less elongated, are shown in cooler colors, and they appear adjacent to the non-branching side of the epithelium that's elongating into the surrounding mesenchyme. So all of this suggests to us that, again, there's this high level of fluidity within the mesenchyme that's taking place concurrent with formation of branches in the bird lung, as we see in the mammalian lung as well. And so from this, we conclude that the forces that induce branching within the epithelium of the chick and the quail and the duck are those actively from shape changes of the epithelium, but then extension of those branches require a change in the mechanical properties, a change in the material properties of the mesenchyme adjacent to those branches. So we're curious to know what induces this active folding of the epithelium, how that pattern is specified, and then we have hypotheses about what's causing that unjamming like transition within the mesenchyme, but we're really interested in understanding both the physical and biochemical signaling that's promoting it. So that's what we know about the forces that are necessary to build the lungs of mammals and birds. In the last few minutes, what I want to tell you is how we think lungs of more, I guess, more primitive lungs form themselves. And this was really inspired by, the really different architectures of the avian and the mammalian lungs, and the fact that per the tree of life, the last common ancestor of these two is thought to be a reptile of some sort. And so this led us to ask, well, how do branches form in the reptile lung? So what does the reptile lung look like? Does it form using a more mammalian mechanism, so with smooth muscle as the driving force and a passive epithelium? Or does it form using a more avian mechanism with an active epithelium and simple fluidity within the mesenchyme? And so this is a project that's really a team effort across multiple groups, but I'm going to show you results that were obtained largely from Katie Goodwin and Michael Palmer. And so to address this question, we established a model system, the anole. So these are small reptiles about the length of a pencil that have a pretty fast gestation time. So they hatch within about 30 days or so, and they have a very simple lung that is known as a faviolar lung. So they don't have alveoli for gas exchange. They don't have parabronchi for gas exchange. Gas exchange takes place at the surface of the lung in regions that are known as favioli. The lung itself is a simple sac. And so I'm going to show you on the next slide a video that goes through the thickness of an adult anole lung that has been stained for the epithelium. And I hope you'll appreciate as the video plays that the lung itself is completely hollow in the center, but it has a really unique structure on its wall. And so that should start playing now. So it's going to loop, but you'll see the left lung and the right lung. Green is the epithelium that has been labeled here. And the surface of the lung is sort of honeycombed in geometry, but the cavity itself is cavernous. And so there's really much less complexity than you see in the mammalian lung or in the bird lung, in the avian lung. And so if you can't see that from the video of the stack, hopefully you can appreciate it from the still images here. So the outer surface of the lung is shown on the left. The inner region of that stack is shown in the middle. And then if you zoom in on that wall, which had sort of like a honeycomb-like geometry, that shows you the faveoli, so the faveolar surface where gas exchange takes place. And the height of those corrugations is indicated, is color-coded in panel B here. So we were curious to understand how such a unique structure can form. And so Michael went and basically carried out analysis of the morphogenesis of this epithelium. It starts out using, looking very similar to the bird lung and the mammalian lung. It's a simple epithelium with a trachea at the top and two bronchi forming sort of a wishbone-shaped structure. And so that's here on the left at embryonic day 5. So E5 means embryonic day 5. A day later, that epithelium has expanded. And so the staining here is for the epithelium, and the lung inflates. So the aspect ratio changes. And then a day after that, so at embryonic day 7, the epithelial surface goes from being smooth to having bumps or corrugations on the surface. And so that takes place over a 2-day period from the wishbone to the corrugation stage. That coincides with a change in the aspect ratio of the lung, which is quantified in panel D. And as that inflation takes place, the epithelium itself changes morphology. It goes from being thicker to a thinned kind of structure, as you might imagine with something that's inflating in a short period of time. And so the cross-sectional area of the epithelial cells increases as this inflation takes place, and the thickness of the epithelium decreases. So then we asked, well, how do these funny corrugations form on the surface of the lung? And it turns out that smooth muscle plays a role here. So again, on the next slide, I'm going to go from left to right, early to later stages. The top row shows you the epithelium again. So we go from a smooth surface at the inflation stage on the left to a bumpy corrugated surface at early and then late embryonic day 7. At the same time as that epithelium is undergoing that change in shape, smooth muscle is forming around this lung, and that's labeled using, again, alpha smooth muscle actin as our marker here. And intriguingly, these corrugations of the epithelium appear in the gaps in between the smooth muscle, which has a really unique geometry in this organ in anoles. The smooth muscle appears like a honeycomb meshwork around the basal surface of the epithelium. And that meshwork appears to guide the appearance of the epithelial corrugations. Now we call this smooth muscle for a couple of reasons. One is that when we take lungs out of the embryo and put them in culture, we can see this tissue actively contract. And so hopefully you can appreciate that from this time lapse, that the smooth muscle meshwork is actively contracting and changing shape of the lung itself. We also come to this conclusion from some single cell RNA sequencing analysis that Michael and Katie did on these lungs. And so, that meshwork geometry of the smooth muscle is really quite striking and unique. And so one of the big questions we've been investigating over the last several months is how that mesh forms. So in order to address this, Michael carried out a number of different experiments. One was just a finer resolution time course looking at the change in shape of the epithelium. Now time is going down on this slide. So the top is the inflation stage, and then the bottom is that corrugation stage of the epithelium, which is shown in the leftmost column. And then staining for smooth muscle is in the second column, and the merge is in column number 3. And then the column on the right shows you a high magnification view of the smooth muscle. So the smooth muscle starts out as basically diffuse over the basal surface of the epithelium, and then refines into these bundles that go across and down the lung itself to form into this meshwork. So it elaborates over time. It doesn't differentiate initially into that pattern. It's elaborated over time. And our data suggests that this elaboration into the meshwork is driven by pressure from the lumen of the lung. And so we come to this conclusion for a number of different reasons, one of which I'm showing you on this slide. If Michael changes the pressure within the lumen, so the pressure of the fluid within the lumen of the lung, he disrupts the ability of that smooth muscle mesh to form. And so he decreased pressure by puncturing the lungs with needles, which allowed the fluid to move out, and the lungs decreased in their volume, which is quantified in panel B. Under these conditions, the smooth muscle doesn't form into that mesh. He increased the pressure in the lung by treating the lungs with forskolin, which promotes the movement of ions and then fluid into the lumen, which increased the volume of the lungs and also prohibits the formation of that smooth muscle meshwork. And so what we think is happening is that there's an optimal pressure within the lumen from the fluid within the lumen of the lung that's setting up stresses that are transmitted into the mesenchyme that is causing the smooth muscle cells to coalesce into fibers downstream of those tension forces, or downstream of them feeling that tension. There's some evidence that this takes place from other systems. But also Michael was able to see it happen in real time. And so this again is an example of epithelial tissues being deformed by virtue of migration within the mesenchyme. And so the anole is not yet universally genetically tractable. And so in order to visualize the formation of this mesh, Michael injected adenovirus that encoded for GFP into the mesenchyme. So it labeled all of the mesenchymal cells and then carried out time-lapse analysis. And that's what I'm going to show you here. So on the left is a control. The GFP labeled cells are on the left, and I hope you can appreciate that they're migrating and coalescing to form these fibers. In contrast, when Michael punctures the lungs and carries out the same experiment, there's much less motility within that labeled mesenchymal population, which, as you saw on the previous slide, never forms into that meshwork geometry. So you might be asking yourselves, is this mesh necessary for the formation of the epithelium and into the corrugations? And it absolutely is. We again have a case where formation of a pattern of smooth muscle is essential for morphogenesis of an epithelium. And we call this stress ball morphogenesis because the morphology of the epithelium and the smooth muscle really look like a contracting stress ball. Okay, so see if I can get this to move forward. The anole airway epithelium looks really similar to the mouse in some ways. It appears to be shaped by physical forces from the mesenchymal population, which is generating a really unique pattern of this meshwork smooth muscle by migration. We don't quite yet understand how growth of the epithelium and differentiation of that smooth muscle population are coupled, or how that meshwork pattern of the smooth muscle is stabilized. But it's a really beautiful illustration, I think, of mesenchymal motility promoting morphogenesis of a sheet. So what I hope I've convinced you by sharing these examples from 3 different species is that there's many ways to take a simple material like a sheet of epithelial cells and turn them into a more complex structure like we see in the vertebrate lungs. The questions that we're really excited to try to answer are what controls these patterns of differentiation? Are there ways for us to control the migration of mesenchymal cells to build epithelial tissues outside of the body? And what other motifs can we discover by looking at additional non-canonical model systems other than the simple lungs of the anole? So with that, the work that I showed you was mostly carried out by 2 really talented graduate students, Katie and Michael. I would be delighted to answer any questions you might have. Thank you.
Thank you, Celeste. That was really great. For everybody out there, just either feel free to do the raise hand function and I'll call on you. And also you can put questions in the chat. So the first question from the chat is from David Shook, asking about what you think regulates the pressure within the lung in the control explants, I guess, for the anoles.
Yeah. So that's a great question. What regulates the pressure within the lung? And so much of our understanding of what controls fluid volume and pressure within organs like the lung comes from understanding of mouse as the model system. And so there, the volume of the fluid within the lung is controlled by both transport of ions from the epithelium and water following passively, as well as opening and closing of the fetal larynx. I have to say, we don't really know as much about development of reptilian lungs. What I just showed you is the first description as far as we know of reptilian lung development at sort of a cellular level resolution. Michael does have single cell RNA sequencing data that includes the epithelial population and is hoping to gather additional single cell RNA sequencing data sets this summer. One downside of the Enola model system is they only breed in the summertime. So this is very seasonal work. And we're hoping that by using those transcript analyses as a guide, we'll be able to pull out the identities of the populations, both within the mesenchyme as well as in the epithelium, and really be able to address that question of do the epithelial cells express the genes one would expect in order to control flux into the lumen. But absolutely, given that pressure seems to be regulating the formation of that mesh, understanding what controls pressure then is one of the next major questions to answer.
Great, thanks. All right, our next question from Jeff Fredberg, if you want to go ahead and ask it.
That was a spectacularly great talk. What an eye-opener. I just really enjoyed that so much. I mean, it's an organ near and dear to my lung, as they say. I had 2 questions. One is, I mean, it's been known for a long time that there are, in the developing lung in utero, mammalian, there are peristaltic waves of smooth muscle contraction going from the central to periphery, thought to be involved in lung development. So my first question is, have you seen anything like that? And what do you think about that? My second question is, in terms of the reptilian model versus the avian model, where are dinosaurs? Because, on that, because some people say that birds are, the last descendants of the dinosaurs. So which way is it?
Yeah, so those are both really fantastic questions. So to your first question, we do see what one would call peristaltic contractions of smooth muscle within the mouse lungs. I think peristalsis in this tissue is a bit of a misnomer. So when I think of peristalsis, I think nice, smooth transition of a contraction down a tissue. That's not really what's happening with the airway smooth muscle. It's less directed. It's more I guess, stochastic, but we do see contractions of the smooth muscle around the airways at these early stages of development in the mouse lung that are, sufficient to promote changes in the diameter of the airways and movement of fluid. How big of a role they play in morphogenesis, we're not yet sure. We did find that in addition to those like less directed, more stochastic peristaltic contractions, there are systemic contractions where the entire smooth muscle surrounding the airways contracts simultaneously on both sides of the lung, so both in the left and the right lung. And those appear to be important for the formation of branches. Your second question, I would love to be able to answer. So you're asking what dinosaur lungs look like. Unfortunately, lungs aren't terribly preservable in fossils. Folks have surmised that the dinosaur lung looks maybe similar to what you would see in a crocodile or an alligator. The adult versions of those lungs, embryonic development of crocodiles and alligators is, I think, even more challenging than our seasonal anole example. We certainly can't have a colony of crocodiles or alligators here at Princeton. I can dream, but I don't think I could convince the animal facility to give us that type of space. But in the adult, those lungs look, somewhat similar to the anole, but more a bit more complex in terms of the numbers of chambers that they have. Michael received a very large batch of chameleon eggs a couple of weeks ago to look at other species of reptiles. And those lungs at first glance look very similar to the anole with some really interesting bird-like features. And so I'm really excited to see what he pulls out of there. I know it won't answer your question about dinosaurs, Jeff, but I think it'll get us a bigger glimpse of what what diversity there really is in lung structures beyond what we've uncovered so far.
I won't go into it now. We should talk a little bit about what sets the pressure in the lung, because we know a little bit about that.
Awesome.
Okay.
Great. Thanks so much, Jeff. I just wanted to say a comment from YouTube. Jody Rosenblatt says, amazing talk. The work is so interesting and beautiful. So I just wanted to pass that on. And then I think the next person with their hand up was Raphael.
Thank you for a very interesting talk. So in mammalian lungs, why only 2 branches occur and not more than that? What defines that number? And another question, in avian and mammalian lungs, what defines the distance between branches? Are there mutants that have different distances between branches?
So when you say what defines 2 branches, you mean why do we have bifurcations and not like trifurcations?
Why it's like trifurcation,
That is an excellent question. I presume that it has to do with the differentiation pattern of smooth muscle. Based on our data, we have not seen trifurcations occurring. That would be interesting to try to maybe induce them. So really test the limits of the mechanism we're proposing by changing the geometry of smooth muscle differentiation to see if we can get that structure to form trifurcations. It could be, physically impossible to do given the geometry of this system. So, from a biophysical perspective, that would be interesting to examine. We really haven't looked at that, but I expect that it's in part based on the geometry of that differentiation that's taking place within that tissue. And then the second question was
distances between branches.
So that's not clear either. There have been some hypotheses that distances are related to basically concentrations of morphogens in the mesenchyme surrounding the epithelium that could arise, say, via Turing type mechanisms. I I cannot yet see how, I guess that would be plausible if those concentration patterns are acting on this tissue that's differentiating around the epithelium, but that hasn't yet been shown. Another possibility is that it has to do with the relative rates of growth of the epithelium and differentiation of the smooth muscle, right? So if the epithelium grows, longer and the smooth muscle differentiates more slowly, then you could change the spacing perhaps. But, we really don't know. I'm sad that I can't answer your question. We don't know.
No, it was good answers. Thank you. Thank you for your answers.
Great, thanks. I will say I've seen Jim Butler make an argument on a chalkboard for a mathematical reason for the lengths, but I can't do that justice, so I won't try. I believe our next question is from Tyler, if you want to go ahead and ask.
Yeah, hey, that null data is really, really exciting. That pattern of smooth muscle is really fascinating. I was wondering if you had looked at the ECM at all yet to see if there's some type of pre-patterned collagen alignment that generates that sort of meshwork that you're seeing there. And also, I was curious if you've looked at all for the, what's in the mouse, what's keeping those subepithelial mesenchymal cells there. In the intestine, Wnt5a has been proposed to be one of these cues and non-canonical Wnt signaling for keeping those cells in close association with the subepithelium.
Both are excellent questions. So Michael did stain for a couple of extracellular matrix proteins. We certainly, when we saw that meshwork geometry of the smooth muscle, a template was certainly like the first hypothesis that we investigated. He was unable to find a meshwork pattern within the extracellular matrix that he was able to stain for that matched the eventual pattern of smooth muscle. That's not to say that extracellular matrix is not involved. I think it probably is, given that he sees that these cells are, assembling into bundles via migration. But we don't have, I guess we have to develop the tools in order to really get at that in this model system. Wnt5a is an interesting molecule in mouse lung development, right? It plays many, many roles. We don't yet have a handle on what it's doing with the subepithelial and submesothelial populations that Katie has pulled out. I think that, given the large amount of motility that she sees in the Dermo-1 Cree confetti mice, many of our conceptual models of, morphogens and biochemical signals within the mesenchyme probably need to be revised, right? So if the cells that are synthesizing and secreting important biochemical signals are actively moving, then what does that mean for these presumed gradients that are somewhat established in our canon of how these organs are developing?
Yeah, great, thank you.
Thanks a lot. And our next question from Heiner.
Okay. Hi, Celeste. Great talk. I have a question to your second part. And is the ventral side of this developing lung not competent to branch? Have you tried some tissue recombination? So, say, dorsal mesenchyme towards the other side?
Oh, yes. I love those classic experiments, the tissue recombination experiments. So we have not. And that is certainly an area that is under investigated by ourselves and others. I don't know whether the ventral epithelium is competent or not. I think that certainly tissue recombination studies would be one approach to investigate that. Another would be to try to do some of these old-fashioned bead experiments where the bead is loaded with molecule known to promote branching. In the bird lung, many of the same signals that are essential for formation of branches that have been found in the mouse have also been mapped in the bird. So I could see doing an experiment where we load a bead, say with FGF10, and stick it adjacent to the ventral surface of the epithelium and see whether a branch can form. We haven't done those in the context of the physical drivers of morphogenesis that we've been investigating. But yeah, that's a great question to try to address. And I think in this tissue, in this context, the mesenchyme recombination experiments would also be feasible. The scale is not too small to do those swaps.
Mm-hmm. May I ask a second question, please? Okay, so have you tried to do single-cell sequencing of the ventral and the dorsal mesenchyme to see the difference?
Ooh.
Okay, it's tricky to separate it, I'm sure, yeah. But...
Tricky, but not impossible.
No.
So we have done single-cell RNA sequencing on the chick lungs. Michael and Katie are putting together comparisons between chick mouse and a knoll as sort of their fun side project. I think that they could probably, even using the single-cell seq data that they've acquired from these stages of development in the bird, if there are differences between dorsal and ventral mesenchyme, they should be able to pull them out. And then we could go back in and map what those cells look like. Yeah.
Okay, great. Thank you so much.
Thanks. Celeste, are you okay to stay on for a couple more minutes? I know we're a bit past time.
Sure, happy to.
Okay, great. We have a couple more questions from the chat. So from Natasha Shiloh, wanting to follow up on Raphael's question, how are the differences in bifurcation regulated in mammalian left and right lungs? For example, there are more branches overall in the right lung.
We don't know. What I was hoping when setting out on this path was that we would find, mutants or manipulations that would allow us to change the pattern of branches, to really get at that question of how this robust stereotypy is controlled. And we've not been able to do that so much. We've been able to find systems that change the rate of morphogenesis. And so we can, slow down development or speed it up or completely block branching. But we have not yet found a system that enables us to finely tune or even grossly alter branch number or position in a way that would get at, sort of the mechanistic underpinnings. I'm sure that some of the answer to that question has to do with the early specification of the overall geometry of the organ. So there's one lobe on the left in mice and 4 lobes on the right. We tend to focus on morphogenesis after that lobar structure has been formed. And I would expect that, much of the answer to that question has to do with, the tricky question of how is the number of lobes specified within a given species, and we don't know the answer to that either.
Okay, great. One thing that sort of related to that, I think this is right. I remember learning that dog lungs have that sort of one branch and then a bunch of branches coming off of it. So and I could be wrong, but that's what I remember. So I'm wondering, how common is this, the bifurcation in mammalian lungs versus other structures?
Yeah, so the bifurcating branching is really what seems to establish that large number of terminal ends that's necessary for having enough structures to differentiate into alveoli. So you have a large surface area for gas exchange once you're born. The side branches in mammals, so those domain branches, seem mostly used, again, I'm anthropomorphizing an organ, but they seem mostly used to generate the overall architecture that is then refined by the bifurcating branches. So the large majority of branches in the mammalian lungs that I've seen, I do not know about dogs, are via these bifurcating mechanisms.
Okay, great. And then another one from the chat. This is a bit long, and I'll try to read it with the right emphasis. We'll see. So lung from Sabag. Lung is intrinsically implied in gas exchanges. It's known that nitric oxide is involved in smooth muscle contraction, at least to regulate blood pressure. So did you investigate the metabolic activity of the nitrogen nitric oxide synthase, PKG activity, etc., or did you apply exogenous nitric oxide to define whether it impacts morphogenesis?
We have not done that. So we have not investigated nitric oxide-based signaling in the airway smooth muscle. We have disrupted smooth muscle contraction. So you can do that a number of different ways. One is to inhibit calcium influx into the smooth muscle. So we use an L-type calcium channel blocker called nifedipine, which blocks contraction of airway smooth muscle. So I think it's similar in spirit to the experiment that you're proposing. That has two effects. One, it blocks the formation of the stereotyped branched architecture of the epithelium in the mouse lung. But the second effect is that it prevents additional smooth muscle differentiation. So contractility of the cells seems to be quite important for permitting them to continue to differentiate.
All right, awesome. Okay, so everybody, last chance to ask questions. And in the meantime, I just want to say, Celeste, thanks so much for coming on. And it was really interesting to hear about how migration is a piece of this, but not all of it. I think some of us who tend to come to this seminar every week, we get really, we really want to focus on that one piece, but it's part of the mosaic here. So that was really wonderful to see. And so I think with that, I'll just say thanks so much for being here. Thanks everyone for coming, and we'll see you all next week.